Thank you to the US FDA for convening this milestone meeting and to the Science Board for your input and expertise. 10 years ago, my poultry science professor introduced me to the idea that we could grow meat from animal cell cultures. As we've moved over generations from extensive to intensive agricultural production systems to create more food from less land, it seemed obvious to me that moving towards highly controlled systems to farm cells instead of animals would be the next paradigm for food technology. And because this particular type of intensification removes the need for whole complex organisms, I suspect that producing meat from animal cell culture rather than whole animals should result in fewer viral epidemics, fewer climate-related catastrophes, and fewer externalized costs to the environment, public health, and animal welfare. While these potential benefits do not pertain to food safety at the level of the individual consumer, they do pertain to food safety at a public health level. With our current production systems, you do not have to consume meat or dairy to be threatened by an antibiotic-resistant infection, an avian flu, or a foodborne illness. Potential benefits aside, there are clearly many unknowns about producing meat from animal cell culture technology, and it is of utmost importance that the hazards, nutritional considerations, and production methods for this technology are well understood to identify risks and ensure consumer safety. I've dedicated my career to this work because I believe this transformative technology was inevitable. However, I want to ensure that it enters society in the most responsible way possible. So it is truly a pleasure to be here today. Before I proceed with answering the 6 questions posed by the agency in today's agenda, I would like to emphasize that there are multiple methods and multiple combinations of methods to produce foods from animal cell culture technology, so that there is likely no one-size-fits-all approach to production, hazard, risk, or safety. It strikes me that existing FDA guidances for existing food and drug manufacturing should provide adequate initial frameworks for assessing the safety of many methods that could be employed to create food products from animal cell culture technology. Question 1, regarding adventitious agents. Yes, adventitious agents could be plausibly introduced into culture from seed cells or culture materials. These adventitious agents could be of microbial, viral, or fungal nature, introduced via the collection of the original tissue, raw materials involved in the cell culture process, or human handling. However, there are already established tools that can be pulled from existing industrial bioprocesses that would be effective in identifying and managing these risks for animal cell culture technology. There is appropriate existing guidance on creating, characterizing, and qualifying cell banks to ensure seed cells are appropriately identified and free of contamination, and I believe that these could serve as the basis for cell banking methods appropriate to animal cell culture technologies. Question 2, regarding the cell culture process. Previous cell culture experience tells us that the potential for contamination cannot be completely eliminated. However, the risk of contamination can be effectively minimized so as not to threaten human health. Established protocols for existing industrial bioprocesses for food and drug manufacturing, such as aseptic technique, sterilization, and routine screening, have successfully minimized contamination risk for existing food and drug products. These existing protocols could reasonably be applied to large-scale production of foods from animal cell culture technology with very low likelihood of risk to human health. Question 3, regarding culture media. Cell culture media are still being developed for all mammalian cell cultures regardless of intended use. In general, we believe that it is very likely that the substances used in cell culture media would not be dissimilar to those used for other human applications, including food. Again, we believe that existing guidances for cell culture and enzyme sourcing can serve as the basis of established operating procedures for animal cell cultures for food production to ensure safety. Similarly, the current advice from the agency on sourcing materials to be used in cell culture media can serve as the basis for developing practices for food production cultures. Question 4, regarding scaffolding materials. Most scaffolding materials would likely be composed of food-grade materials such as cellulose, collagen, or chitin. These materials could be absorbed by the growing cellular matter or could be intentionally removed from the final product and could easily be evaluated using existing food ingredient evaluation procedures to ensure safety. We note that novel scaffolding materials could create metabolites or residues in the cell culture process that remain in the finished food product. Such novel scaffolding materials, which could change in the culture process, may require safety evaluation that does not fall under existing food ingredient evaluation procedures and will likely require food additive approvals prior to use. Question 5, regarding errors in the culture process. For large-scale culture of a single cell type, there are existing protocols and assays for detection of errors in the culture process. The likelihood that harmful substances such as metabolites or allergen would be created are quite low. However, in the absence of peer-reviewed research on large-scale culture of all meat and seafood-relevant cell types and species, it's hard to conclude definitively. Further, there is a gap in FDA guidance around co-cultures or the simultaneous culture of multiple cell types and/or cell species. In this scenario, it would be important to understand how multiple cell types may interact in culture conditions. I do not believe there are existing frameworks for evaluating the safety of co-cultures across food or drug manufacturing, but New Harvest welcomes the opportunity to work with the agency and other experts in developing the background science to identify potential risks and hazards. Question 6 regarding nutritional considerations. Nutritional considerations have always been a thorny area for evaluation. Because the fine composition of food from animals or plants differs based on genetics, husbandry, and food sources, performing these analyses are often dependent on selecting the appropriate comparator. We note that the animal cloning risk assessment provides an excellent methodology for determining the nutritional sufficiency of meat and milk. We encourage the agency to look to that paradigm for safety and nutritional evaluation. It is very possible that meat produced through cell culture technology could exhibit a wide range of nutritional value, largely determined by the composition of the cell culture media during proliferation and differentiation of the cells in culture. I believe that it would likely be that the nutritional profile of a meat produced from cell culture technology could be designed to be similar to conventional meat, either through manipulation of the cell culture media or through enrichment and fortification processes taking place post-harvest. I would be curious to see if the bioavailability of certain nutrients would differ if they were incorporated into the product via the cell culture process compared to via post-harvest enrichment. For example, will iron be present in the heme or non-heme form in a final food product? In summary, I believe that there are already frameworks in place from food and drug manufacturing that could assess and manage the risks of a cell-based meat manufacturing process. However, these existing frameworks come from both food and drug manufacturing, and there may be differences in the intended use and the route of exposure of the products evaluated by these existing frameworks and the intended use and route of exposure of a cell-based meat. Further, while the frameworks may cover several processes for cell-based meat production, I'm not sure that they can cover all future processes, given that there are so many opportunities for novel innovation in the development of cell-based meat production processes. The aforementioned co-culturing of multiple different cell types is an example of an innovation where I have not seen prior guidance or frameworks for evaluating safety. I have one additional point of note. The basis of good governance and good regulation is informed by evidence and peer-reviewed research. Federally funded research is an important part of this equation. Perhaps because of the novelty of this research, or perhaps because it pulls from expertise historically created in medicine but instead applied for food production, this research has not received meaningful federal support. The majority of research in the production of cell-based meats has come from venture capital-funded companies or from philanthropy-funded research organizations like New Harvest. These funding streams have definitely brought the field to this point. However, for this technology to be fully realized, not just as a product, but as a new paradigm for food production, we will need to see more support for academic, peer-reviewed research. Perhaps the identification and support of research initiatives that would both equip regulators and minimize burden on innovators could be a good, important way for the FDA, USDA, and perhaps the NIH and NSF, etc., and the burgeoning cell-based meat industry to work together. My organization, New Harvest, as a primary funder of academic research in this space to date, would be a keen collaborator and resource in moving this forward. Thank you to the FDA and the Science Board for